Where's my stick? Okay. I have to have my prop. Okay, today we're going to talk about coevolution. And this is another one of these topics that has a long history, especially a long history of very indirect and tortuous development within phylogenetics or phylogenetic comparative biology. So today this is going to be a mess, but don't be afraid. We have a plan. And one of the biggest changes between the 1991 book called Phylogeny, Ecology, and Behavior and the 2002 upgrade of that called The Nature of Diversity, one of the biggest, the chapters that changed the most was the coevolution chapter. I mean, basically, that chapter was in the 10 years between 1991 and 2002, that chapter was completely restructured. And Deborah McLennan and I often refer to that chapter as the Stockholm chapter because most of the reason we were able to completely restructure that chapter was because during the 1990s, Deborah and I went to Stockholm University every autumn and taught a portion of their evolution course. And then we would attend a conference that they had at that time every year. And we got to talk to a lot of really interesting, really intelligent people and ended up interacting with 3 different academic generations of people. One of the people that I met, Sura Nulin, who I met first when he was a graduate student, is now the chairman of the department. That will either make you feel proud or make you feel old. I'm sorry, classic. Okay, so coevolution. How complicated can it really be? I mean, you know, it's fairly simple. It's just a word made up of 2 parts. There's co, meaning together, implying that we're dealing with more than one species, and evolution, which means descent with modification. And in modern days, coevolution has come to mean any hypothesized or postulated interaction between 2 species in which the species or the groups of species are expected to have modified each other through time. So it's come to mean mutual descent with mutual modification. Now it turns out that the mutual descent and mutual modification parts aren't necessarily that closely tied to each other. And the assumption has always been that they are. And this is the point at which I entered the arena of discussion in this particular area. I published a paper in 1979 in Systematic Zoology where I looked at 2 different aspects of host-parasite associations from a phylogenetic standpoint. One was this issue of whether or not host and parasite phylogenies were congruent with each other. In other words, does the parasite phylogeny map the host phylogeny? If you took a parasite phylogeny and replaced the names of the parasites with the names of the hosts, was that just another way of showing the host phylogeny? And then I also looked at, or tried to look at, phylogenetic aspects of everything that we call host specificity. And the first thing I realized was that they weren't the same thing. In other words, you couldn't determine from whether or not the host and parasite phylogenies were completely congruent or incongruent. You could not determine just from that what kind of host specificity you might be dealing with. And the other aspect was also true. You couldn't just look at host specificity and automatically assume that you knew whether or not the hosts and parasites were associated through phylogenetic time. You couldn't say anything about the matching of the phylogenies. And so the aspect of coevolution that we call mutual association, I called that co-speciation. And the aspect of mutual modification, I called that co-accommodation. And what I said was that these are 2 different aspects of parasite evolution. but they aren't necessarily connected to each other in the sense that phylogenetic patterns here don't necessarily tell you anything about this, and phylogenetic information there doesn't tell you anything about that. So patterns of co-speciation, and by co-speciation I meant mutual association. Co-speciation in the strict sense is that matching of host and parasite phylogenies, but the whole area of mutual association also included phylogenetic patterns of host switching. And in 1981, I came up with a method that Ed Wiley later called Brooks parsimony analysis that was an attempt to try to document very closely the points in a parasite phylogeny that were congruent with the host phylogeny, and even more importantly, the very specific cases in which the parasite phylogeny did not match the host phylogeny. So there was an emphasis on detecting episodes of host switching in that methodology right from the beginning. Now that method treated parasites and their phylogenetic relationships as being analogous to characters of the host. Okay?
Okay. So that's a little piece of history. We put it off to the side for the moment. Now, what do we, meaning people who study coevolution, tend to assume? Well, we tend to assume that the number of hosts and the geographic range of the parasites are proportional to their degree of ecological specialization. We tend to assume that. Widespread species, we assume they're generals. Species that occur in many hosts, we assume that they're generals. And we tend to assume that the probability of host switching, of being able to move from one host to another, is inversely proportional to the degree of specialization. So, the more specialized you are, the lower the chances of being able to host switch. What do we observe in nature? Well, we observe that most species of parasites or associates, that is, insects that feed on plants, hymenopterans that are parasitoids on other insects, anything you want to call a parasite in a very loose sense, in nature, have a small number of host species and a small geographic range. Therefore, we assume that most parasite species are extremely specialists, and therefore host switching is a rare phenomenon, and most of the parasites can be treated as if they were inherited with the hosts, as if rather than being analogous to host characters, they really are host characters. And this led to the theory of maximum co-speciation. Now, here we've got this very complicated terminological history because the word co-speciation is used in three different ways in the literature. There was the original way that I intended it when I first made up the term, which was this just has to do with phylogenetic studies of mutual association. There is this sense of co-speciation, which parallels the theory of maximum vicariance, and that says that parasites are characters of their hosts. Just like in maximum vicariance, species are characters of their geographic areas. And this then, of course, leads to an application of the methods in cladistic biogeography, where instead of having geographic areas, you put the names of hosts. But it's still the same operation. Now, the historical origin of that particular theory, even though you don't see it in the literature until the early 1980s, but it actually, under that name, but it actually has a much longer history. It's not a new concept. In the late 19th century and early 20th century, it was sometimes called the parasitological method. And the parasitological method is simply using parasites as characters, taxonomic characters of the host, and assuming that the parasite relationships were determined by the host relationships. So this leads to some very common practices in parasite taxonomy. Among others are the practice of, oh, I have a parasite in this species of host. No one's ever reported this parasite group in this species of host. This must be an undescribed species. I will name it. Which, of course, reinforces the whole theory. Now, in 1993, okay, now Hennig attacked the parasitological method in 1966 well, actually, in his 1950 book, and the 1966 English translation, which is where I read it. And basically what Hennig was saying was, we have no way of telling when a parasite is an apomorphic trait of the character. We treat parasites like characters. We don't know when a parasite should be an apomorphic and when it should be a plesionomorphic. And of course, the thing that he was neglecting was that parasites have their own phylogenetic relationships. So in 1981, when I first published the first paper on BPA, I called it a solution to Hennig's parasitological method. Because the way to determine whether a parasite has an apomorphic or plesiomorphic relationship with its host is to not just include the presence or absence of the parasite, but also include information about the phylogenetic relationships of the parasites, put it into a historical context. In 1993, when Deborah McClellan and I published the book Parastrict, which was Historical Ecology for Parasitologists, it sold 6 copies, which tells you, how interested and how aware of modern systematic practices parasitology was at the time. But we were able, in this book, to do a little more detailed historical analysis. And what we discovered was that this idea that hosts and parasites have the same phylogeny doesn't come from any kind of Darwinian or even neo-Darwinian or even Lamarckian thinking. It comes right out of orthogenesis. And the idea is, in orthogenetic theory, you have these independent origins of life, progressive specialization through time of independent lineages, until you get to a point that you're so specialized that you become completely dependent on something else, that is, you become a parasite, and from that point on, you have no evolutionary independence. You are a reflection of whatever taxon you're dependent on until you cause your own extinction by becoming so overspecialized you can't survive. So the expectation among orthogeneticists was that hosts and parasites ought to have the same phylogeny, because they were, by the time you got to be a parasite, you had no independent biology of your own. You were, in fact, simply a passive characteristic of the host. So, for the maximum co-speciation program, and that's the same people who were doing cladistic biogeography were doing this. The methodology that you want to develop is one which finds the maximum degree of congruence between host and parasite phylogenies. And it turns out that you can use these old methods, and you can use assumptions 1 and 2 to eliminate any data that look like they support host switching. Because we know, quote unquote, we have already assumed that host switching is extremely rare because of the high degree of ecological specialization in the associations. Therefore, anything that looks like host switching is really just incomplete data or misleading data, and we can fix those bad data with assumptions 1 and 2. And what you get is a way to modify any parasite data into a reflection of the host phylogeny. So in this sense, you are not treating parasites as analogous to host characters. They are host characters. So it turns out then that that particular line of thought, that tradition, especially, you can see it especially well when you start to look at both parasitology and insect-plant studies side by side in a historical sense. What you find is that there are 2 very old traditions. There are 2 traditions of looking at what we now call co-evolved systems. They go back to the late 19th century. And one of them we call the host-centered studies. That is, the primary in the investigation is the host. And in those studies, the host species is the evolutionary resource. That is what the parasites are specialized on, the host species. And this is the tradition in parasitology. Now, the tradition in insect plant studies is an insect-centered view, or a parasite-centered view, which basically says the host species has something that is the resource. And if some characteristic of the host species is the actual resource that the parasites that are using in maintaining their association. The reason for the association is not because of the host species, but because of something the host species has. And that was the basis of the part in my 1979 paper where I talked about co-accommodation, talked about host specificity and things like that, where I said, if the host has a critical resource, that resource can be apomorphic or plesiomorphic, it can be convergent, it can be anything, and therefore you can have any kind of association between the host and parasite phylogenetically, and you cannot tell just from the degree of host specificity what's going on. You can't explain the evolutionary reason for this, the context. So here's a little bit of comparison. All right. This is a phylogenetic tree, and let's say that we've got 4 different groups. This is 4 different species of parasites, and here's a host phylogeny. Now, host phylogeny has been generated by independent characters. So you have, here's the parasite phylogeny, here's the host phylogeny. Now, if you simply replace the numbers representing parasite species with the letters representing host species, this diagram and this diagram are identical. And the same thing is true. We've seen this pattern before, right? The same thing is true here, where what you have is a host, host B, which does not have a member of that parasite group. Now, because the rest of the diagram, the rest of the phylogeny of parasites is congruent with the host phylogeny, we just assume that there was a parasite in host B that's gone extinct, or we just haven't found it yet. And this is a situation in which you have lineage duplication, blah, 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 blah. So these 3 simple modules of pattern produce a diagram of the host phylogeny based on the parasites that looks just like the host phylogeny based on anything else. So if all evolution of host parasite associations represent one of these 3 outcomes, then you in fact will always see the host phylogeny and the parasite phylogeny being congruent with each other. The problem is that there are a lot of ways in which hosts and parasites can be associated phylogenetically that do not mirror the host phylogeny. And they produce more complex patterns. So unless you're going to say that none of this exists, you need a method that doesn't oversimplify the data. Now, if you only get this complexity view by taking the perspective of the parasite, and some of the people in Curso and some of the people here, parasitologists that I've been speaking to the last few weeks, know that one of the things I keep saying is stop thinking about the host, start thinking about the parasite, stop thinking about the host. Now, it's difficult, for example, if you're trying to get funding. If you're trying to get funding to do parasitological research, it's much easier to get funding if you're studying parasites that are important to hosts that are important. So if you say, I want to study this group of tapeworms, you're not likely to get funding as well as if you say, I want to study the parasites and the potential impact of these parasites on this group of commercially important fish, or this group of fish that are important for sport fishing and ecotourism. So you have to be sort sort of split in two. You have to, and on the one hand, you have to understand that the people who have the money to support parasitological research think about the hosts. But you can't make the same mistake. You have to understand that you will never understand the parasite biology properly if you take the perspective of the host. You have to take the perspective of the parasite. And that means that if the parasite has the resource, if the host has the resource, so the host is just carrying a characteristic, then you can expect to see any of these. You can see co-speciation or extinction or sympatric speciation of parasites, but you may also see speciation by host switching, and you may just see expanded host range as a result of host switching. That is, adding hosts without speciating. And you'll recognize that this is the same thing as the widespread species phenomenon, or the widespread species problem. So here's the host phylogeny, there's the parasite phylogeny. Guess what? Right? Okay. Here's some parasite that's gone off and colonized some new host and messed up our nice co-speciation diagram. Here's a case in which you have two species of the parasite group in the same host, but not for the same reason. Not because of sympatric speciation within the host, but because of one episode of say, co-speciation of origin, and a second episode of speciation by host switching. Now we know that this is speciation by host switching because A and D, host A and D, are not sister species. And here you have the widespread species phenomenon. Here's one where the parasite's found in more than one host. Okay. So it turns out that this host-centered view, this traditional parasitological view, is very much in alignment with the simplistic or the simple view of the universe. This Aristotelian Platonic view that the world is very simple and essentially static. So it turns out that this is one of the problems with these kinds of simplistic views is that they're derived from a static ontology of the universe. So the attempt to make a static ontology into an evolutionary research program is always very difficult. It always creates problems. And this is what we do with the sort of the maximum co-speciation program. We only allow co-speciation, extinction, and sympatric speciation. And we just eliminate host switching because we've decided that it's very rare. Now, among other things, obviously, if you can convince policymakers that evolutionary biology says that host switching is rare enough to be treated as an error term in models, then we should not be spending any extra money at all on trying to anticipate and adapt to and deal with emerging diseases, because the evolutionary process says it's very unlikely that there are going to be very many emerging diseases. Now, I already talked about this last week. The parasite-centered view is complex. It is more in alignment with the complexity view of the universe, the evolutionary view of the universe. And it allows these processes, but these as well. And it says that because various kinds of host switching are possible, and in some cases maybe even quite frequent, we really have to include host switching as part of the explanation in our models. Host switching can't be considered an error or a problem to be corrected. It has to be part of the explanation. Now, both of these traditions agree that parasites are ecological specialists. Okay. It turns out that's almost the only thing they agree on. But everybody, quote unquote, does agree that parasites are ecological specialists. And they are specialized in two primary ways, again, that everybody agrees on: microhabitat preferences and transmission dynamics. and everybody agrees that whether you're talking about Phytophagous insects or tapeworms, these things are highly specialized in terms of these elements of their ecology. Now, traditionally, we have used host range, the number of different hosts utilized, number of different host species utilized, as a token of this, because this is more difficult to document. And this is very easy. So we can collect those data very easily, and if the number of host species utilized is an indication of how specialized or how generalized you are, then why not do something really quickly? Now, there are two fundamental questions that pertain to this. One is there much opportunity for host switching? Are we correct in assuming that all this ecological specialization precludes very much host switching? And then, is there much opportunity conceptually? And then, the more empirical question, which is, what do we see out there in nature when we look at the phylogenetic relationships between parasites and their hosts from the perspective of the parasite evolution? So this means that we actually have to stop thinking about the co-speciation part of coevolution and phylogenetics. We actually have to spend a lot of time on the co-accommodation part, the host specificity in the big sense. And the first is, conceptually, what are the opportunities for host switching? Or another way of putting that is to ask this question: how can extreme resource specialists host switch readily? And you've seen this before a couple of times. Here you have a host phylogeny, and here this thick line represents one host with one parasite. Now this is the way parasitologists, at least, tend to think most of their world is structured. That most of the world is structured in such a way that you have one host that is the only survivable host for the parasite. And there are a lot of different models of coevolution, but all of them have the effect of making this relationship more and more and more specialized, to the extent that we tend to think that if this host goes extinct, that parasite goes extinct. Now, if it turns out that this parasite is not specialized on the host species, but rather specialized on something the host species has, a characteristic of the host species, but this is the real evolutionary context, then another way of saying this is that the parasite is specialized on an apomorphy of that host. So the parasite is locked in on a real inherited trait of the host, but it's an apomorphy in that host species. Now what happens if that trait is in fact not an apomorphy for the host species, but a synapomorphy for a group of host species? Well, that means that if you find this parasite inhabiting all three of these hosts, it still has the same evolutionary relationship. This and this are situations in which the parasite is equally specialized. There is no difference between those two diagrams in terms of the quality of the specialization. Because as far as the parasite is concerned, all of these hosts are the same thing. Only human beings recognize the hosts as being different species. But as far as the parasites are concerned, they're all the same thing. And of course, that means, among other things, that if the parasite originated only with host G in an isolated geographical area, but for some reason moved into the area where this host or this host lived, we would predict that that parasite would instantly begin to be associated with those other hosts. Because as far as the parasites were concerned, they weren't moving into the neighborhood of a new species of host. They were just moving into a place where there was more of the same kind of habitat. as their original host, because it's all the same resource. And if the resource is a persistent, even more ancient character, persistent plesiometric trait, you still have exactly the same degree of ecological specialization in the host-parasite relationship. Except now, not only do you have multiple hosts, but those hosts don't form a clade. They're not, quote-unquote, closely related. But this doesn't make the parasite a generalist. In fact, this diagram, and this diagram, and this diagram, the parasite is equally ecologically specialized in every single case. And it also even holds true if the trait, the resource, the host resource, is convergently evolved. So it turns out, this is a really fundamentally important observation. It turns out, as I said in 1979, it turns out to be true that you cannot predict how much host switching there has been by looking at the number of hosts, by looking at host specificity. Because host specificity has nothing to do with the host species. It has to do with the resource requirements of the parasite and how those resources or that resource are distributed phylogenetically among all the possible hosts in the world. So if that host resource is phylogenetically conservative, the parasite can be extraordinarily specialized and still have a wide range of potential hosts that it can use. So it can not only host switch readily, but it may have a big host range that would mislead some people into thinking it was a generalist. So that's the problem with host range. If you count the number of host species, you're treating the host species as the resource. And the assumption is that each host species, because it's a different species, is a different resource. So if you're in 10 different species of host, you must be a generalist. If you're in one species of host, you must be a specialist. Plus, at least in parasitology, and it's probably not that too much different when you look at all of insect taxonomy, but at least in parasitology and helminthology, the vast majority, something like 75 to 80% of all of the named species, are only known from the original description, which means one host, one place, one time. And in Parascript, Deborah McLennan and I were able to find a couple of examples. There are not very many in the literature because parasite taxonomists just, there aren't very many of them. They're not funded very well, they're not supported very well, and there's an overwhelming amount of diversity to try to get through. But in the few cases that we've been able to find in the literature where you can take a group of parasite species and you can do just a simple linear correlation analysis between the number of hosts reported for the species and the number of times that species has been reported in the literature, the correlation coefficient is about 0.9. And I've said this before. Somebody asked me a while ago, after 15 years of doing a parasite inventory in a conservation area in Costa Rica, they said, what do you know? After 15 years of this massive inventory, give me your, the take-home message. What have you learned? What are the great insights? And I said, the only thing we know for certain is that every story in all of our textbooks is wrong. We know that's the case. And it's especially true with host range, because every single species of parasite we collected in Costa Rica that had been named before was found in a host that had never been listed as a host before. All of them. So our sense of host range for most of our parasitic worms is just an artifact of bad taxonomic inventory work. Now, but that doesn't mean then I'm saying that all the parasites are actually generalists, and we only think they're specialists because we haven't looked for enough hosts. Just because we find these things in additional hosts doesn't mean that they've suddenly become generalists, or that they were generalists all along and we just didn't know it. Because whether you're a generalist or a specialist cannot be determined just by counting the number of hosts that you live in. All right, now, we believe, if you look at parasites from the perspective of the host, that specialization on the host species mitigates against host switching, so that through time, as the host-parasite relationship becomes more and more specialized, the chances of host switching become less and less and less. Now that means, among other things, that from this point of view, evolution is the best safeguard against emerging disease. The appearance of host switching is an artifact of incomplete information about lineage duplications and sorting events, or sympatric speciation and extinction, or it's the result of not using assumption 2 enough in massaging your data so that it looks just like the host biology. So it's reasonable to use these assumptions to get the answer that you already know is true. The parasite-centered view simply says, over long periods of evolutionary time, host ranges expand and contract, and expand and contract, and expand and contract. Now this pattern has been documented really well by the Stockholm group for butterflies, for lepidopterans. A big, huge lepidopteran tree of life, you plot the number of species of host plants on that tree, and you know, this big, huge, deep phylogeny, and what you find is the host range just goes wah, 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 wah. And that tells you that if an increase in host range is associated with things becoming generalists, and a decrease in host range is associated with things becoming specialists, then, through evolutionary time, you have both specialists becoming generalists and generalists becoming specialists. But you also have to remember that just because host range expands, that doesn't mean that something's becoming a generalist. So what that does is it forces us, this so-called oscillation hypothesis, forces us to look more closely at exactly what is the relationship between host range and the evolution of the insects or the parasites. So you have to look at it from the parasite point of view. And it turns out that if there is phylogenetic conservatism in the traits or trait that are carried by the host, that are the resource that the parasite zeroes in on, then you have a lot of potential complexity in terms of things like host switching and host range. Now host range, not only can you have things with big host ranges that are specialists, as well as things with big host ranges that are generalists, you may also have things with narrow host ranges that are generalists, but are looking like specialists because there are other specialists in their environment that are preoccupying resources as hosts that generalist could use under other circumstances. So this is what Deborah McClellan and I, big host range, which you're still a specialist, we call those faux generalists. Little host range, but you're actually a generalist because of the local ecology, we call those faux specialists. We also have an additional element to this that the Swedes in Stockholm have documented, and these are things called polyspecialists. So they have actually done experimental work to show that there are some butterflies that have very specialized associations with multiple hosts, but the association is different in each case. So they're not generalists, they're not just using any old plant out there in the same way, but they're highly tolerant of the differences in plants. What they have is a very specialized association with each of a collection of plant groups. So they're actually polyspecialists. And for the parasitologists, think of digenetic trematodes, for example. Digenetic trematodes are a classic example of polyspecialists. At one stage in the life cycle, you are extremely specialized on a mollusk. At another stage in your life cycle, you may be extremely specialized on a fish, or an arthropod, or vegetation. And at another stage in your life cycle, you may be very specialized in a bird. And when you're in the mollusk, you're in the hepatopancreas. When you're in the second intermediate host, you're in the muscles or the mesenteries or the exoskeleton. And when you're in the definitive host, you're in the intestine or the lungs or something like that. So trematodes, every species of trematode is inherently a polyspecialist. And they carry within their genomes different partitions that allow them to express different kinds, very different, qualitatively different kinds of host specificity at different stages in their lives. That's within one individual, never mind species or group of species. Well, this is ecological fitting. And ecological fitting is what allows specialists to host switch, and it's what allows specialists to have big host ranges, and being able to host switch and having big host ranges is a necessary part of becoming a generalist. So ecological fitting is what allows specialists to exist really well during periods when we would think that being a specialist would be very costly. And it's also a way for specialists to produce generalists, so that being a specialist is not an evolutionary dead end. Now, it turns out that ecological fitting, Dan Jansen coined that term in 1969. And in 1976, John Maynard Smith coined a term called diffuse coevolution. And in 1979, I coined this term called co-accommodation. Now, it's all the same thing. So why are there three different terms? Well, it's because Dan Jansen was an ecologist, Maynard Smith was an evolutionary geneticist, and I was a taxonomist. And we didn't read each other's literature, we didn't talk to each other, we didn't go to each other's meetings, so we didn't know about each other's work. And this is just a classic problem with over-specialization in our professions. Because this really is over-specialization. We lost 20 years here by not talking to each other and not realizing we had a common concept. Okay. Now, one of the corollaries of ecological fitting, as I've mentioned several times now, is Sal Augusta's concept of sloppy fitness space. That just because you're extremely specialized, it doesn't mean that your association with your host is optimized in any particular way. And in fact, for most parasites, where there's a very low probability of any one parasite propagule finding a host, maintaining host-parasite associations through time is such a chancy proposition that there really isn't much possibility for any kind of genetic reinforcement that's going to lead to any kind of optimality. And that's not true for all situations. And it's not true in local contexts. You may have local situations where you have a very highly predictable, very intense kind of interaction. And this is what John Thompson calls a coevolutionary mosaic. So that at the population level, you may have these very tight kinds of coevolutionary arms races or resource trappings or whatever is going on. But at the macroevolutionary level, those kinds of phenomena do not get translated into patterns of host-parasite associations, macroevolutionary patterns of host-parasite associations, because there is a relationship between being specialized and the conservative nature of most traits, that is, the conservative nature of most resources. That always means that no matter how tight the specialization becomes in one local area, globally, regionally, or globally, there's still an enormous amount of unoccupied, usable fitness space. So can specialists host switch? Well, not if the host species is the resource. But if the host has some resource, yeah, you can be extraordinarily specialized and still be able to host switch easily when circumstances change. So have we, and by that I mean the maximum co-speciation research program, overgeneralized by forcing all the data on host parasite phylogenies to match each other. With very sophisticated methods, very complicated algorithms now, to simplify the data, simplify the parasite data. And is the cost of producing those simple explanations greater than the cost of allowing complex explanations? If you have to minimally postulate at least one lineage duplication and one extinction event for which we have no evidence, rather than allowing one host switching event for which we do have evidence, are you not producing non-parsimonious explanations in order to maintain a simple result and to support a simple theory? And this is where I get to introduce one of my favorite American authors, Mark Twain, and this is from Life in the Mississippi. He said, there's something fascinating about science. One gets such a wholesale returning conjecture for such a trifling investment of fact. And what he was talking about there was he was making a parody of the scientific method, which in the 1870s was still a fairly new phenomenon. And so he said, all right, I'm going to apply the scientific method to the evolution of the Mississippi River. Now we know that as the Mississippi River goes through time, it winds around and curves around, and then occasionally, in one of those curves, it actually cuts through. And when it winds around, the river gets longer and longer and longer, and when it cuts through, it gets shorter and shorter. And so he says, now by applying scientific rigor, I can say that when the Mississippi River arose, and he said, in the Devonian, which was 1 million years ago last Thursday, it was 1 inch long. It was 2.5 centimeters long. He said, and 500 years from now, because it's twisting so much, 500 years from now, it's going to stretch out into the Gulf of Mexico like a fishing pole, because it will become so long. And he said, and that's when he said this. He said, you know, look at all this amazing imagination you can get just by thinking about a little tiny bit of fact. And that's in reality what has happened with a lot of these thoughts about maximum co-speciation and the methodologies. The people who are doing this primarily are people that don't have an enormous amount of practical field experience with host parasite systems. The people who have real experience out there with insects and plants, or with tapeworms and catfish, don't find that to be a very satisfactory maximum co-speciation, to be a very satisfactory explanation for the world. So, now let's go back and start looking at, and we've seen this before, now we start talking about the so-called cost of specialization. Because we have this enormous amount of theory that says that it's really expensive to be a specialist. And that's based originally on the idea that, well, if you're an extreme resource specialist, eventually you're going to become so specialized that you're going to go extinct. So if you're specialized on one host species and the host species goes extinct, you go extinct. If you are so specialized on one resource and that resource disappears, you go extinct. The perfect example of something like that is the giant panda. The giant pandas are specialized on bamboo. And any botanist will tell you that one of the things that's amazing about bamboo species is that they don't flower very often. In fact, there are some species of bamboo that we have known about since the 19th century and no one has ever collected a flower. There's some species of bamboo that have not flowered in the 150 years since they were discovered by Western scientists. The other thing is that when bamboo species do flower, they die. There's a massive die-off for the year. So here you have the giant panda. Giant panda specialized on a food source that periodically dies for a year. Think that might have some bearing, might be part of the reason why giant pandas are an evolutionary dead end, why they are the opposite of an evolutionary success story. And maybe World Wildlife shouldn't have put all that money into saving a dead end species. That's another issue. But the idea then is that what you've got, and this is why Maynard Smith got interested in it, because Maynard Smith was always promoting the idea that everything that happens in evolution is a combination of costs and benefits. So you never have a situation in which something new arises and it's beneficial and the old thing is costly, so the old thing goes away and the new thing sticks around. Rather, what you have is everything has costs as well as benefits. And as long as the benefits outweigh the costs, whatever it is will continue to exist. So the cost can be great, but if the benefits are even greater, then you continue. Or the cost can be very small, and the benefits can be very small. And whatever it is will continue to survive. So the idea about the relationship between generalists and specialists is that specialists are supposed to be better at utilizing resources, their particular resource, than generalists. So if you give a specialist on rice and a generalist who can eat rice the opportunity to use rice, the specialist will outcompete the generalist for that resource because the specialist is specialized on. That's the thing. Also, so that means that when the environmental conditions are very predictable and very stable, specialists should be at an advantage over generalists. On the other hand, when the environment changes, generalists should be at an advantage over specialists because if you're specialized on something that's very restricted, the environment changes, that resource goes away, you die. So the idea is that over evolutionary time, you should have periods of time when generalists are favored, then specialists, then generalists, then specialists. And by now, we should be living in ecosystems that are made up of about equal numbers of generalists and specialists. And this will allow us to predict that the next time there's a big climate change event, the generalists will survive and the specialists won't, and that's how we're going to replace the biosphere. Now, ecological fitting says, well, the cost of being a specialist isn't necessarily that great. Because if you live in one area and you're specialized on a certain host resource, and the climate changes and you leave that area, you may move to an area where there's another host that has the same trait because of conservative evolution. Now, you don't have to become a generalist to survive. And your original host may go extinct. And this is one of the things that Eric Hoberg, one of the most fundamentally important set of observations that Eric Hoberg has made, is recognizing that there are a number of parasite lineages that are older than their hosts. And the only way you can find a parasite lineage that's older than the host is if the parasites have survived host extinction events by just moving from one host to another. Now, normally we would say, oh, well, the only parasites that can do that are generalists, but these are all specialists, and we don't have any evidence that they've ever been generalists. But it turns out that ecological fitting is a mechanism whereby a specialist has the same flexibility for adapting to new conditions as a generalist. So that means that the cost of being a specialist is not as great as we thought. And so even though we observe this at any one time slice in nature, we go out right now, most parasites right now have small geographic ranges and only occur in a small number of places. That does not mean that they are limited in terms of their ability to survive major climate change events. So the phylogenetic conservatism lowers the cost of specialization because you can host switch without becoming a generalist. You can host switch without having to evolve new capabilities for utilizing the host. And then we also have all these other things that I talked about a couple of weeks ago, these indirect effects. There's ecological context. You may be capable of inhabiting multiple hosts in your environment, but because some of those hosts are preempted by other relationships, or because there's something about your biology that doesn't allow you to come into contact with those hosts, you simply don't see them. It's what the insect plant people call appearance. So a butterfly can fly into a field, and if 99% of the plants in that field are one species and only 1% is the preferred plant species for the butterfly, and the field is large enough, the butterfly may not have time to find the preferred plant before she has to oviposit. And as a consequence, the preferred plant is there, but it's not apparent. And so the eggs get deposited on a plant that's okay, but it's not preferred. You know, all kinds. We won't worry about these indirect effects. Well, I really do, not just because we get 75 cents in royalties for each one bought, but I really do encourage you to buy copies of The Nature of Diversity. It's actually, buying the paperback is cheaper than photocopying it, because it's like over 500 pages, it's only like 20 bucks from Chicago. Okay, so specialists, it's not as costly to be a specialist as we once thought. And it turns out that it may be more costly to be a generalist than we thought. Okay? So remember that we're thinking about specialists being at an advantage in terms of utilizing particular resources over generalists. Now, the only time the generalist would be at an advantage is when the specialized resource went away. And then the specialist would be at a disadvantage. But if that disadvantage isn't there, so if you have, say, a change in environmental context, and the specialist can still survive really well by being a specialist, that never gives the generalist an opportunity to be at an advantage. So it turns out that, in fact, instead of talking about the cost of being a specialist, we probably should be talking about the cost of being a generalist, and ask why there ever are generalists. And it may be that when we see this expansion and contraction of host range in the oscillation hypothesis, the expansion of host range and the production of generalists may actually be evolutionarily short. That may only happen for a very short period of time, because there may only be very, very specific times when it actually is advantageous to be a generalist. And part of it is because you end up, as you add more and more hosts and more kinds of hosts, you increase the chances that you may make a mistake with what kind of host you get into. You then start to have things that we call preference hierarchies and things like that. And it becomes very complicated, very interesting. But the bottom line is, it turns out that the more complex the ecosystem, the more costly it is to be a generalist. So, when is it good to be a generalist? This is really weird. This is a very cruel Darwinian paradox. The best time to be a generalist is when you don't need to be a generalist. That's very Darwinian. Neo-Darwinism says you should be a generalist only when it's best to be a generalist. You should be a specialist only when it's best to be a specialist. But it turns out that in Darwinism, the time to be a generalist is the time when the environment is stable enough so that evolutionary experiments can happen. See, if you're a Lamarckian, you wait until the crisis happens, and then you evolve the proper adaptation to survive. You made it again. If you're a Darwinian, and you wait until the crisis happens, you go extinct. So the only time to experiment, the only time to produce non-optimal, but positive fitness variants is when they're not necessary. And the Swedes have done, the Stockholm guys have done some brilliant experiments about what happens if you have a butterfly that normally only has one chance to oviposit each year. They're extremely choosy when they only have one chance. But in years when they have two chances, they're very choosy the first time, but the second time, they mess around. And usually, if the next year's, you know, a bad year again, most of those things don't work, but that's okay because they've already produced one set of offspring. Okay? So when the environment's really very particular, very harsh, there's a very narrow window of time, they don't experiment. That's exactly when evolution, Darwinian evolution, does not produce novelties, is when the environmental crisis is there. The only time Darwinian evolution produces novelties is when you don't need them. But that's because in Darwinism, fitness space is sloppy. It's not survival of the fittest. It's survival of anything that has non-zero fitness. It's survival of the active. Relative cost. Faux generalist, faux specialist again. All of this, these are just, now the reason I'm running through these is because Walter's going to be converting these PowerPoints to text and then putting them on the website so you can download this stuff. But these are just little sound bites from the coevolution chapter in the nature of diversity. And you really need to, if you're doing any insect plant work, if you're doing any host parasite work, you really have to read that chapter and then look at the literature that's cited there, because there's enormous background literature to this. And most parasitologists are completely unaware of any of this stuff. And yet, we've got systems that are really tractable, really good for looking at some of these very general things. Okay, now, if it turns out that there's a cost to being a generalist, there's not so much of a cost to being a specialist, that the time to be a generalist is when you don't need to be a generalist, then there's one more factor in that, and that is if you are a generalist, and being a generalist is not so advantageous, what's the best way to survive if you're really a generalist? This is the converse of when is it advantageous to experiment when you don't need to. The best way to survive as a generalist, if being a generalist is costly, the best way to survive as a generalist is to act like a specialist. Now, if you're a generalist, but there's one resource that's underutilized, spend all your time with that. It doesn't cost anything. The environment changes, you still have the ability to use other resources. And in that case, your ability to use multiple resources makes you just as survivable as a specialist on phylogenetically conservative and widespread resources, so you won't be at a disadvantage. So any faux specialist that is a generalist that is restricted in its ecosystem to a small number of resources is going to be acting like a specialist and is going to enjoy the benefits of being a specialist. Now let's talk about counting hosts and deciding who's a specialist and who's a generalist. If there is one indicator of whether you're a specialist or a generalist that is absolutely misleading, it's counting hosts. And that is the way the vast majority of people, ecologists, distinguish generalists and specialists. And it's the worst measure we can use. The ecological fitting invalidates any notion that the number of host species is a direct indicator of whether it's a specialist or a generalist. And sloppy fitness space implies that no matter what you see right now, the species always has more options if the environment changes. Either as a generalist acting like a specialist, or as a specialist on phylogenetically conservative resources. So, it's possible for extreme resource specialists to switch easily and often. It's possible for generalists to appear to be specialists, which means they're also able to switch easily and often. Therefore, there's no reason to assume that host switching is rare. And whether you can host switch or not is independent of whether you're a generalist or a specialist. The reality is we should expect host switching to occur often. And that means that when we look at host and parasite phylogenies, we cannot use methods that eliminate everything that doesn't conform to the host phylogeny. And of course, there's a sidebar to that, and that is, if you're using the host phylogeny to manipulate the parasite data to get the right answer, that means that when there's a discrepancy between the parasite phylogeny and the host phylogeny is always correct and the parasite phylogeny is always wrong. So what are the odds that all those vertebrate people doing cytochrome B trees are always correct? And the parasite phylogenies based on 7 different genes are always wrong. Just saying. Okay. Important conclusion. There are no phylogenetic patterns of host-parasite relationships that can distinguish among the models of coevolution. Any of them. Now, in 1991, in phylogeny, ecology, and behavior, Deborah and I had these elaborate diagrams. This is what resource tracking would look like. This is what an arms race would look like. All these very complicated diagrams. And some of the guys in Stockholm were working really hard to try to figure this out as well. And I remember we arrived in Stockholm one time because we'd been messing with this, and we'd come to a certain conclusion. And I remember arriving in Stockholm, one of the first things that Seward and Newlin said to us that time was, you know, we've been looking at this and we've decided that there just aren't any particular phylogenetic patterns. And we just pulled out a bunch of papers and said, yeah, we came to the same conclusion. So we have to rewrite that. If we ever do another edition of that book, we have to rewrite that entire chapter. And that's right. Host switching or no host switching tells you nothing about the quality of coevolution that's going on. It doesn't even tell you if coevolution is happening. So we have these two sorts of issues. One is that you can't use traditional means. The traditional means we use for determining specialists and generalists are wrong. Phylogenetic comparisons between hosts and parasites don't tell us about coevolutionary dynamics. And because host switching can be really easy under whether you're a generalist or a specialist, we can't use methods of comparing host and phylogenies that preclude host switching. But we need information about the host-parasite associations, the phylogenetic context of those host-parasite associations, to understand any of this stuff. So, because our keys to understanding coevolution are in the host switching events, not in the co-speciation events. The co-speciation events are default. They tell you nothing except that the parasites and the hosts were together through time. It's host switching that tells you something about what's going on. Okay, now, 15-minute break. Don't worry, we have a plan. We'll come back, and what I'm going to do next is I'm going to show you a bunch of examples. Well, some examples. We don't have as many actual published examples of host and parasite patents in the literature, there are one or two examples that people just keep re-examining and re-examining, and there's not a whole lot of new studies out there. But I'll show you what we've got, and then we'll loop back into what does this mean and what do we do with it. Okay. See you at 10. It's 9:44, right?
9:44, yes.
So we'll loop back at 10 o'clock. Okay, we will get started again. Okay, just to recapitulate where we finished. Sound okay? Okay. Comparisons of host and parasite phylogenies are not explanatory. It doesn't matter how much agreement or disagreement you have between host and parasite phylogenies. That is not going to tell you anything about the actual evolutionary, ecological, or coevolutionary interactions that are going on. You need to do other things for that. If you really want to do that kind of work, then John Thompson's most recent book is the book you want to buy. Now, however, the fact that these are just descriptions doesn't mean that they're irrelevant, because the descriptions are really important as part of explanations. And in particular, we really want information about the host switches and patterns of host switches. So, now I've gone through this whole theoretical thing about why host switching ought not to be expensive, ought not to be rare. We should expect host switching to happen a fair amount. And now what I want to do is to say, okay, if we begin to analyze parasite data using methods, comparing the parasites with their hosts, using methods that allow host switching as a possibility, what does the world look like? Okay, when we use original methods of historical biogeography, the whole world looked like Vicarians. Once we start using methods allow a full description of the actual evidence, the entire world looked like taxon pulses. Okay, and I'm not going to show you some of my earlier work using BPA, which made the world look like it was all host speciation of host and parasite phylogenies. But I will go through a couple of examples, including one that is a reanalysis of my dissertation work and produces a completely different story, which is much more interesting than I came up with in my dissertation. So, now, one of the things that I've already mentioned is that the logic of the methods that we use for comparing host and parasite phylogenies is the same as for biogeography. So, you just think of hosts as pieces of geography, and the relationship between the parasites and the hosts is the relationship between species in the areas where they live. So here's something that we find very commonly in parasitology. This is a phylogeny for a group of tapeworms called Taenia. This is one of our robust studies. And this whole thing is color-coded for different kinds of mammalian hosts. And what you find on these is a really interesting situation, because there are obviously, a lot of host switches going on. And in this particular case, what Eric was most interested in was, where do the three species of human Taenia come from? So the three species of Taenia that infect human beings. Two of them are here, and one of them is here. Okay? These two are related to, most closely related to a species of Taenia that lives in a cat. And this one, Taenia solium, is most closely related to a species of Taenia that lives in a hyena. And this is African and this is Asian. So you can tell a lot from these patterns. Okay, you don't know what the exact coevolutionary dynamics might be, but you do get a sense of what was going on. Okay, and we know we have really good evidence here for the relationship between humans and hyenas. There's a paleontologist at the Natural History Museum in Stockholm named Lars Perelin, who has done this really amazing analysis of all of the non-hominoid fossils in the Olduvai area, where the Leakey Foundation has done all of the ancient hominoid research. And Lars has this really densely documented, really well documented sequence where there's an association between hyenas and humans beginning 1.5 million years ago. And 1.5 million years ago, the hyenas were the predators and humans were scavengers. 1 million years ago, they were competing with each other. And by 500,000 years ago, the humans were the predators, the hyenas were the scavengers, and that's what we have today. So today we teach that hyenas are scavengers, but they're only scavengers because they were pushed into being scavengers by human beings who transitioned from being scavenging primates into being top-line predators. Well, this is part of the cost of human beings becoming predators. When we become predators, we get to the infected prey first, and we get infected. So this is how these host switches happen. This is one of the costs of civilization. You know, we only think about civilization as being this wonderful benefit to humanity, but the reality is, between becoming predators and then developing domestication and developing agriculture and then living in cities, human beings have acquired an enormous number of diseases, especially parasitic diseases, as a result of our increasing levels of civilization. So this is a perfect example of the fact that everything that happens in evolution has costs as well as benefits. Now obviously, the benefit of being a predator outweighed the costs of having these new tapeworms. But that doesn't mean that having the tapeworms is good for them. It just means that that's what happened. So you can take something like this, and if you want to understand the places where there's been an evolutionary change, where something interesting has happened, it's in the host switches. This is where the interesting stuff is happening. Here's like a canid, a dog to a hyena. Okay, dogs, cats, hyenas, and then into humans. All these sorts of things. There are a lot of different potential scenarios, and this also gives us insight into the evolution of life cycles and so on, transmission dynamics, and possibly things like where exactly in the hosts they live. So this is a really good example. All of the really interesting evolution that you can tell from this comparison between hosts and parasites comes from understanding the host switches. Now you've seen this example before. This is a packed analysis. There's also a secondary BPA analysis in the literature of these two groups of nematodes, one group of hookworms and one group of pinworms. And the pinworms, this group of pinworms, is one of two groups of parasites. And the last thing we'll do today is I'm going to walk you through a reanalysis of what's now considered to be the classic case of co-speciation. But the other one that's supposed to show, these guys are supposed to show a one-to-one relationship between the host and parasite phylogeny And in fact, this is one of the groups that was originally used in developing the parasitological method. This is one of the groups that was used 50 years ago, 60 years ago by parasitologists to say, look, each of the great apes has its own pinworm, and from that, and therefore the pinworm phylogeny is the host phylogeny, and therefore all pinworms are like this. So you fast forward from about 1930 to the 1990s, when I started up this parasite inventory in Costa Rica, and where we collected something like 45 different species of frogs, and we collected pinworms from something like 30 species of frogs. Okay? Now, the expectation is that means there are 30 species of pinworms. And probably all of them are undescribed. So what was the reality? The reality was that there were 7 species of pinworms, and one of them is in 23 different species of frogs. So I'm just saying, these are pinworms. What we have traditionally thought about them and what the reality is don't necessarily match up. So here's the pack diagram. This is not a host phylogeny. This is a branching diagram and summarizes the combined histories of 2 parasite groups in the context of their hosts. And it's not a host phylogeny based on parasites. We don't really care about the hosts. We're trying to explain the parasites. And hosts are just one ecological variable in the evolution of the parasites. The red lines represent places in which the parasite data agree with the host phylogeny. And all the green lines are cases of host switches. In this particular case, it's about 40% of the host associations are due to host switching. And about 60% are due to common association. And the reality is that these episodes of common association, they don't tell us anything. They just say the hosts and the parasites were hanging out together for 30 million years. The host switches, on the other hand, tell us a lot. And one of the things they tell us immediately is that with the exception of this host switch, all the rest of these host switches are switches between species within that host group. So these are just recombinations of host relationships among the group of hosts that were already infected, which means that this is probably all just ecological fitting. And instead of looking for new genes for getting into new hosts, what we should be looking at is changes in geography and climate and ecology to explain how, for example, baboons or macaques and orangutans might have ended up sharing parasites, or how could gibbons and macaques end up sharing parasites, because gibbons are really arboreal. I mean, they really never come to the ground at all. And macaques and things like that, if any of you have ever seen them, they're not up in the trees very much. They can go up in the trees if they have to, but they're mostly on the ground. So there's some interesting stuff here. How do you get these things that are down on the ground to host switch with things that are up in the trees? And well, of course, the obvious thing is here, it's from the things that are in the trees to the things on the ground. Okay? The explanation there is probably just gravity. The enormous amount of the larval parasites and the eggs of the parasites in these guys don't stay up in the trees. So then you might say, oh, okay, well, if it's true that these guys picked up a parasite by host switching from the things that are up in the trees, because the things that are up in the trees are crapping on the ground, then you turn the question around and say, okay, if they're always crapping out of the trees down on the ground, how do they keep the parasite cycle going up in the trees? Okay, well, this is the point at which parasitologists have no trouble discussing it, and everybody else in the world goes, I don't want to hear about it. So, parasitologists will talk to you. All right. This is abdomen, of course. This is a bonobo. Probably our closest relative, even though we tend to classify both species of pan together. It may actually be that the pan meniscus is our actual sister species. Okay. Blah, blah, blah. Now, all but one of the host switches involve the original host group, so that's almost certainly all ecological fitting, which means changes in geography, changes in ecology that bring susceptible hosts into contact with parasites they've never seen before. All right. There is one case that involves a switch from primates to rodents. Okay? And that may have, I mean, those are distantly related organisms, that may have required some kind of evolutionary innovation on the part of the parasites. If that's true, then that's the one case, that's the one place you want to go and really study more closely. Okay, so if you wouldn't want to waste your time studying any of the other species to try to find evidence of a genetic change associated with a host switch. Okay. Now, all that ecological fitting, all that host switching by ecological fitting, another way of characterizing that is to say that this is just the parasites wandering through sloppy fitness space. As the opportunity arises, they just move into new pieces of fitness space that were not previously available to them. Okay. Now, ecological fitting, this phylogenetic conservatism, creates lots of uninfected hosts that are nonetheless susceptible all over the world. So a parasite originates in one place, specialized on one host in that one ecosystem in that one place, but there are a whole bunch of hosts in other ecosystems that represent the same resource. And if the parasite ever gets to one of those other ecosystems, it will just move into that new host because it's not new resource. It's old resource in a new place. So the phylogenetic conservatism in host resource evolution, coupled with speciation by geographic isolation, produces this macroevolutionary mosaic on the planet where the non-zero fitness space, all of the host space on the planet where any given parasite could conceivably survive is really large compared to where the parasite actually is at any one point in time. And then all you need are changes in geographic range and or trophic interactions to produce instantaneous host switches. And all host switches are what we would call emerging diseases. Okay, so there is a prediction that comes. If what drives host switching is anything that changes geographic distributions and trophic structure of ecosystems, and by that we mean climate change, primarily global and regional climate change events, then host switching should occur in bursts in time. Host switching should not be something that happens stochastically. Little here, unpredictably, randomly, as unpredictable, random genetic changes that produce the capabilities to infect new hosts arise. Okay, now that model of random genetic evolution for the ability to attack a new host. So I think that is another kind of magical thinking. And it's based in part on the perspective that parasites, the function of parasites is to attack things. That's where we get this coevolutionary arms race mentality. Parasites are trying to do something bad to the really valuable organisms on this planet, which are the hosts. Okay? Now, no parasitologist has that perspective. This is all the perspective of people who are trying to explain the parasites through the filter of the hosts. But from the filter, the perspective of the parasites are just trying to produce the next generation. And in fact, parasites are really happy when the host doesn't even know their name. You know, if you can hang out inside the host and the host immune system doesn't even know you're there, this is great. The host isn't spending energy making antibodies, and you're not having any trouble with an inflammatory response from the host. Everybody gets along just fine. Okay? So, all the host switches that Eric Hoberg has documented in his studies, and he published a big review article. He was kind enough to let me be a co-author on it, but it was his article in the Journal of Biogeography a couple of years ago, which is this huge review. And every single case that we've been able to document, there are bursts of host switching, and those bursts of host switching are associated with major climate change events. And in between those climate change events, there's no host switching. And what happens with those climate change events is they're like, oh, expansion, and then isolation. Expansion, and then isolation. So what we see is that these bursts of host switching are tied to the biotic expansion events in the taxon pulses. So one of the things that happens when climate change perturbs the environment and changes circumstances, some species move. And two things happen when those species move. One possibility is that they bring parasites, their own parasites, into contact with new hosts that have never been exposed but are susceptible because of ecological affinity, or they move into a new area without their original parasites, but they meet parasites there that can inhabit them easily because from those parasites' perspective, these new hosts moving in are just the same phylogenetically conservative resource. So one of the things we expect is host switching in both directions, and host switching to occur easily and quickly, but only under two kinds of circumstances. Big changes in geographic distributions of hosts and or parasites, and changes in trophic structure. So the only thing that prevents massive numbers of emerging diseases from occurring is that human beings don't have massive population expansion, so that human beings are moving into new areas where they've never evolved. Yeah, I guess we do that, don't we? All right. Well, another reason we're not going to have lots of emerging diseases is because human beings are not moving species all over the planet and introducing species into new areas where they've never been. Oh yeah, we do that too. All right. Well, really, the main reason we're not going to have a lot of emerging diseases is because there's no climate change. Oh, yeah, there is. The whole world is an enormous minefield of potential emerging diseases. And the way to access those is through human population expansion in areas where humans have never been before, introducing species into places they've never been before, and climate change, which changes the geographic distributions and trophic structures for whole ecosystems. And as long as any or all of those things are happening, and they're all happening right now, the prediction is that we are at the beginning of one of these bursts of host switches, which means we're just at the beginning of an outbreak of emerging disease. Charles Darwin again is saying, hey, biogeography, woo! distribution of hominoids since the Miocene. Here are the parasites of the hominoids. Okay, not surprising because most of these are extinct, and we don't know anything about their parasites. But as you can see, the parasite distributions do coincide with that scenario. So now we can go back to that path diagram for the parasites and say, what was going on with the parasites? So now it's not just the elephants and the hyenas and humans, it's the elephants and the hyenas and humans and the human parasites. They're all doing the same thing. And look what this is. Africa to Asia, Africa to Asia, Asia, Asia to Africa. And over here, Africa back to Asia, this is us. This is Africa, this is out of Africa, into Asia, over into North America, and so on. This little bit here is what we did. That's why there's some great, I mean, the parasitologists here will already know this, but some of the, well, not some, the best paleoparasitologists in the world are Brazilian. And there's an incredibly interesting story that's been reconstructed about human introduction, humans introducing themselves into South America, and the parasites they brought with them. And they brought parasites in multiple ways. There are parasites that have been brought to Brazil with the slave trade. There are parasites that were brought to the southern United States from Africa with the slave trade. There are old parasites from Asia that have come into South America. There are young parasites from Asia that have come into South America. There's one species of hookworm that appears to have been introduced to South America about 600 years ago when the Japanese fishing fleet was washed away from Japan by a storm or something, ended up coming ashore on the coast of Peru and establishing a village there where they lived for a generation or two and then died out. But not before they managed to transmit their hookworms to the human beings who were already there. So, and all of those, if there had been doctors around the time, they would have, oh my God, there's this emerging hookworm disease outbreak in Peru. What's that all about? We better do some massive sequencing of those hookworms to find out how coming to Peru created the new genetic material for inhabiting people in Peru. And then we would have spent enormous amounts of money sequencing things like crazy. And we would have put more and more money into sequencing because the more we did not find differences, the more we would be convinced that they were there and we just needed more sequencing. When the actual explanation was really simple. And it was ecological and evolutionary, not genetic and pharmacological. And now about 1/3 of all the host associations in that particular example of the hookworm and pinworms and primates, about 1/3 of the associations are due to host switching. Now, that may not seem like a lot, but considering that this was supposed to be an example of no host switching, that's quite a bit. And there are other published studies that show actually quite a bit more. Most of the host switching happens in conjunction with episodes of global climate change and the biotic expansions and alterations and autotrophic relationships. Okay, so now I've been talking about real parasites and real hosts. What about insects and plants? Okay. Well, it turns out that we can see exactly the same pattern. And this is a genus of beetles that feed on leaves and their host plants in North America. And this is a summary of a whole series of studies that Doug Fortuna and his students did. And here's what you've got. There's a phylogeny for the beetles. Go to host plants. Okay, now the short thing is, here's the story. In North America, before the last ice age, you have plants all over North America, and there are beetles all over North America. And the beetle species are isolated in very particular places, and the plants are all isolated out. And then the glaciers form, and everything gets pushed farther south. And they get pushed together. So a lot of beetles and plants living up north get pushed down south, and beetles and plants all come into contact with each other, and everything gets all mixed up. And then the glaciers go away, and the plants and the beetles start to move out again. Except now, all of the beetle and plant associations are different from what they were before the glaciation. They're still extremely specialized ecological associations, but because of the glaciers pushing them together, it's all been mix and match. And then they expand out, and it's like by magic, there are all these new associations. And that's what this shows. And everything that's in purple here, those are host switches. I mean, this is all host switching. And those are all host switches among close relatives. This is all ecological fitting. This is just, they're pushed together, they mixed up, and then they expanded out again, and it looked like it was a completely new ecosystem. You do have a couple of cases where some interesting things happen. Here are some host switches into distantly related plants. So during that time, you did have some funny things happen. And this one time, this is a big change. But here's the thing. You have this big change in terms of the host group, colonized. But after that, it's all ecological fitting. So you have a very small number of true innovations and a lot of ecological fitting to generate this incredible amount of diversity. And the real driver of that, of course, was just climate change. Okay. Oh, okay, right. It's getting late in the week, so I have to remind myself. Where did we go? All right. Now, as promised, this unfortunately has not been published yet. This was a study that was done originally as a class project by two of Bill Fahn's students at Southeastern Louisiana University. And Sarah Temple, not Sarah Brock, no. Sarah Brock, and she's something else. Sarah Brock, that's her name. So all of us had sort of these reticulated histories of multiple marriages and things like that. Sarah is now teaching at a junior college in New Orleans, and every so often I write to her and say, "You ever published this? You ever published this?" It hasn't gotten published yet, but I'm really hoping that we can get this out, because this is a reanalysis of my dissertation using PACT instead of BPA. And it is, and in my dissertation, I only looked at the trematodes of crocodilians. This is the trematodes and the nematodes of crocodilians. And of course, it's updated for species that we didn't know about back in the Pleistocene when I did my dissertation. Okay. So, and this is a lot of this is redundant because this is part of, this was the PowerPoint they put together for their class presentation. So, types of parasite evolution, types of patterns, okay. Different discovery methods, BPA and PACT. And this is assumption 0, right? Here's BPA, this is blah, 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 blah. This is what it used to look like. These are the matrices you used to have to produce to do BPA. Because you had to turn all of the parasite trees into binary matrices. And then you had to do that. That was a mess. And if you had missing data, then there were lots of question marks. This is a mess. PACT makes that much simpler. If I can ever get somebody to actually do the program. All right. Now, there were 2 papers by Poe and Gatesy that expressed unhappiness with BPA. Now, the reality is that what you need to know about these 2 papers is that these were papers about crocodilian phylogeny. Now, in my dissertation, I did this primary BPA and I said, oh look, the parasites seem to be giving us the host phylogeny. Well, it turns out that some of the relationships in the host phylogeny suggested by primary BPA, these are not host phylogenetic relationships that the crocodilian specialists actually agree with. So there was a problem. They wrote these papers and they said basically, BPA is not giving us the right host phylogeny, therefore there's something wrong with BPA. But the reality is that BPA was originally formulated in order to highlight host switching events. And if you have a lot of host switching, if you have any relationship that's primarily due to host switching, you would expect BPA to very efficiently give you host relationships that are incongruent with the host phylogeny. So we're sort of arguing at cross purposes here. I mean, what they're saying is, we don't like BPA because it doesn't turn the parasite data into the correct host phylogeny. Okay? It's not supposed to. And besides, by 1996, we had secondary BPA and phylogeny conjugator and parascript, where there were multiple things in literature saying, no, primary BPA is no good. So, you know, when they found that primary BPA was no good, that was not news. We already knew that. So the fact that, you know, they didn't get the right host phylogeny is not actually a big shock. However, as we now know, PACT is better than BPA, so we don't worry about it. And now remember, this is for class. So, you know, they had to explain how you can actually turn a phylogenetic tree into a Venn diagram straight. So, this is just a comparison between them. So here's the parasite phylogeny. This is their little pact thing. You replace the names of the parasites with the names of the hosts. And then you start analyzing. Now, what clade parasites, only clade parasites. And so these two combine goes back. And these things that are highlighted are actually the only places that were common. Right, and you know about this methods, combination methods, combination rules. Okay. Now, two groups, roundworms and trematodes. Now, this is part of the story. One of the things that's always concerned me about crocodilians is that I wanted to do the phylogenetics of crocodilian parasites for my dissertation because this is the mid-1970s, late 1970s. That was a period of time when there was a lot of argument about whether or not phylogenetic systematics should be banned from biology because it did not recognize a taxonomic group called reptiles. Now, Claytis kept insisting that crocodiles and birds should be classified together, and that mammals were the sister group of everybody else, and not the youngest mammal, all this sort of stuff. So I said, okay, if crocodilians are actually the closest living relatives of birds, then everybody who's done comparisons between crocodilians and other reptile parasites have missed. They've been doing the wrong comparisons. So I'll look at the crocodilian parasites specifically with an eye towards finding out these parasitological connections between crocodilians and birds. It turns out there are almost none. And in fact, crocodilians don't have that many parasites compared to birds. And if you go, for example, if you go into the Paraná where we were, and you kill a lot of the herons and egrets and other water birds, you'll find an enormous number of species of tapeworms and acanthocynins. No crocodilian anywhere in the world has a tapeworm or an acanthocynid. There are maybe 15 different families of trematodes in aquatic birds. The crocodilians have about 4 families represented. So this is actually the real story of crocodilians. And I was just focused on trying to find a connection, but the real evolutionary story is really, why don't they have more than they have? Because, you know, the jacqueries here are eating the same fish infected with the same larval trematodes and tapeworms that the garces are eating. And none of those parasites are maturing in the crocodilians. And, you know, those crocodilians have been living side by side with those birds for, what, 50 million years, 100 million years? And they're not exchanging parasites? I mean, that's an interesting story. There's a really interesting story there about why the crocodiles don't have more parasites than they do. And we're not going to understand that from this. We're going to understand something else. Okay. So, again, this is... All right. So this is the host tree based on the nematodes in crocodilians. And then this was just to show them assumption 1. So this is one of the clades of nematodes, another clade of nematodes, another, okay. This is just to show you that assumption 0 is always satisfied. Okay. So there's your final tree. Now, this is what we get. Oh, look. Lots of parasites in the same hosts, speciation by colonization, rapid exchange, range expansion of parasites, so the same parasite, many hosts. Everything that's like this is co-speciation. So all of these double lines are places where the host and parasite phylogenies agree. So it turns out there's 6 nematode clades, and about half, a little bit more than half of them coincide with the host phylogeny. Okay, now, if you are a crocodilian phylogeneticist, 50% is not good enough. Okay, but if you're a parasitologist working with assumption 0 and ecological fitting and oscillations and taxon pulses, that's actually quite stable. So one of the things we begin to see here, at least with the nematodes, is there may not be very many nematode groups in crocodilians, but the groups that they got have a long, stable association with them. Once they got into crocodilians, they didn't do very much that was very adventurous at the time. But there are 9 reticulations as multiple species in the same clade of parasites in the same host. And they involve a whole bunch of different hosts, and they account for 64% or 2/3 of the host switching. And all of the parasites in Crocodylus, Niloticus, and alligator are the result of host switches. And there are many dispersal events that include parasite species colonizing new hosts or recolonizing old hosts, so producing reticulations. Okay. This is what I got with the old primary BPA. Okay. And you can see why the crocodilian phylogeny people wouldn't be very happy with those results. And one of the reasons there was all that ambiguity was because I was using a method that was forcing the data to oversimplify into something that was as close to the host phylogeny as possible. Okay. Now, we've got a whole bunch of digenean groups. And this is what the host phylogeny looks like for the trematodes, all the trematodes combined. And you can see there's a mess. I mean, there's a lot of ambiguity. And either that means we just haven't looked at very much, or it means something, whatever that point in time was, all hell broke loose. That there were a lot of things were moving around and stuff was switching all over the place. Okay, and again, you see, and in this particular case, if you keep looking here, what you find is that big polytomy is not because there was a big burst of everything, it's because there was a burst of something involving the host, and the parasite groups did not uniformly participate. There are a whole bunch of different variations there. Everything is sort of a subgroup. Here you got, okay, speciation by host switching. Lots of what looks like sympatric speciation really rapidly. Lots of host switching correlated with environmental change. Well, they found that, in fact, that was associated with a major climate change event. This, in fact, is probably the KT boundary, where lots of crocodilians went extinct, and the ones that didn't go extinct moved around. And various bits and pieces of the parasite group survived in various hosts. So everything was sort of fragmented up, but a relatively small number of crocodilian hosts survived, and a larger number of the parasite group survived than that host survived. And you end up here with the digenes. And remember, the digenes were the ones that I was doing in my dissertation, supposedly showing all this massive amounts of co-speciation. But in fact, if you use PACT, you find out that only 17% of those relationships actually are in agreement with the host phylogeny. Now, why did primary BPA find something that was close to the host phylogeny when only 17% are like that? Well, it's because the host switches, even though 83% of the associations are host switches, you never have correlated host switches that outnumber the co-speciation events. So it's just by default that you ended up getting something that looked like host phylogeny, but it was not. It's just, it's an artifact, and it's certainly not the most interesting thing about this. What's really interesting is, and here, see that they equate co-speciation and coevolution, which is something a lot of parasitologists do, and we know it's not. But here it says that most of the speciation in the trematodes is speciation by colonization, speciation by colonizing new hosts. And remember, most of the action in that diagram is out of that one big burst, which is one big climate change. That's exactly what you would expect. Right at climate change, you have all these host switching events because of geographic expansion. And that's when you would get all of the new hosts being colonized. And subsequent to that expansion, you would have a period of isolation, and you get lots and lots of species that all came out of that one burst. But they were all host switching events at that point in time. Now, it's not 227% of the cladogram is host reticulation. What they were actually, this was a bit of a joke. What they're saying is that the number of hosts and the number of hosts, there are more than twice as many host associations as there are hosts. This is just a little joke that they were using. So, but what they're basically saying is that most of that diagram is reticulations, which is consistent with the idea that most of it is the result of this massive expansion under climate change, in which you have hosts and parasites dispersed, and everybody's trying to survive somehow. And you end up, in a way, it's the opposite of the beetle picture, where you have the glaciers compress everything and they mixed up, and then they just expanded out. This is everything exploded out, and you had mix and match as they were exploding out. And that's the big deal. And this is just, you this stuff. That's why we need pattern analysis, blah, blah, blah. If anybody wants bibliography, we can do that. Okay. Now, back to, all right. Now, we're going to take a 15-minute break. And I'm going to draw the host cladogram, the host phylogeny, and the parasite phylogeny for the classic case of co-speciation on the board. And when we come back in 15 minutes, I'm going to walk you through it. And I'm going to show you that if co-speciation means that the host and parasite phylogenies are congruent, then the classic case of co-speciation is only supported by 1/3 of the data. However, there is a way, we have a plan, there is a way for it to be most speciation by host switching and still be co-speciation in the sense of the host and the parasite speciate at the same time. But it's not co-speciation in this sense of the host and parasite phylogenies being congruent. It's something else. Okay, back in 15 minutes. Give me 15 minutes to draw this up on the board, and then come back, and we'll go through this. I know, I should have done it before, but Okay. I can add it later, yeah. That's all right, it's just, it's more hands-on to do it this way, yeah. And lice. Pocket gophers. Now, this is being called the classic case of co-speciation. Okay? It was originally published by Hafner and Adler in Nature, and they claim in that this was the best documented case of co-speciation ever found. And since then, it's been picked up, even though in phylogeny, ecology, behavior, and parascript, and the nature of diversity, the Deborah McClellan and I said there's a lot of post-switching in there. And despite the fact that Eric Hoberg has published saying there's a lot of post-switching in there, it has become the classic case. It's like Rosen's classic case of vicariance. This is the classic case of co-speciation. And it's now found its way into textbooks. Okay? So this is in, for example, it's in Ridley's evolution textbook as this is undoubtedly co-speciation. And it's actually, this diagram, a better version of this diagram, is actually put in there, and then in the figure legend it says, host and parasite phylogenies mirror each other. Okay. Now, these red circles that I have drawn here are places where large chunks of the host and parasite phylogeny do not match. And the red lines that are drawn here are the 3 cases of post-switching that were actually identified in the original study, but they said, I just won't worry about that. So if you actually go on and just count up the number of matches and mismatches between the host phylogeny and parasite phylogeny, what you find is that if you take the outgroups out, you have 1, 2, 3, 4, 5 matches with the host and parasite phylogeny, and 1, 2, 3, 4, 5, 6, 7, 8 mismatches. So, what? It's like the pinworms and primates. It's like between 30 and 40%. A third, 40% is co-speciation. The rest is all speciation by host switching. Now, why did they make the mistake? Well, one of the reasons they made the mistake was that except for these 3 cases where you have a parasite species in more than one host species, all the rest of these parasites are only found in one host. That means they're really specialized. That means they can't host switch. That means the parasite phylogeny ought to be congruent with the host phylogeny. Okay. Now, in addition to that, if this is co-speciation in the sense that the hosts and parasites speciated at the same time, we should be able to find that the molecular clock estimates for the age of these speciation events should be the same for the hosts and parasites. And they are. Well, they are in a sense. They are when you correct for the differences between the parasite molecular clock and the host molecular clock. So in the raw data, if you use one clock, the host and parasite ages don't match up. But if you do the proper correction, they do actually match up. Okay, so let's assume that that's right, because they were correcting basically for generation time, assuming that the parasite generation time was much greater. Okay, so you do, if the parasites produce 5 generations in the time and the host produced 1 generation, you just adjust the molecular clock estimates and you assume that the parasites are diverging faster than the hosts. I don't really have too many problems with that, because it's actually kind of cool if it does turn out that the parasite speciation events and the host speciation events are the same age. Because there's only one way that can happen. There's only one way you can have no co-speciation or very little co-speciation in the sense of host and parasite phylogeny matching up, and still have host and speciation events occurring at the same time. Okay, so co-speciation means the host and the parasite are the same age. Then, with their particular adjustment of the molecular clock, this is all co-speciation. If co-speciation means the host and parasite phylogenies are congruent, this is mostly not co-speciation. So there's a paradox. It is or it is not co-speciation. Now, if these are supposed to be host and parasite phylogenies mirroring each other, like Ridley says in his book, then it's a lie. It's simply not true. And anybody, I can teach any undergraduate how to optimize trees in 15 minutes, and they can do the same thing I just did and show it's not true. On the other hand, if you want to change the definition of speciation so that you're just saying that host and parasite lineages are the same age regardless of how the association was established, that's okay. Just say, I'm changing the definition of the term, and then understand that you've got something very interesting. Because, like I said, there's only one way for this to be co-speciation. Okay? Now, standard speciation by host switching is like peripheral isolate speciation. Remember, peripheral isolate speciation, the separation, whatever the discontinuity is, has to be older than the speciation event. So standard speciation by host switching, the host species is already there, and then the parasite colonizes it. So you expect the parasites that speciate by colonizing, every one of these, the parasites should be younger than the hosts. Unless the reason for the host switches was an episode of climate change, or episodes of climate change, in which the hosts were also moving. So that you had hosts and parasites dispersing, and as that happened, the parasites were finding new hosts and jumping to new hosts. And so the hosts were speciating as a result of their own peripheral isolate speciation. At the same time, the parasites were establishing new host-parasite relationships as a result of ecological fitting. So the same episode of climate change as a forcing event could actually produce a complex history in which the host group and the parasite group were both actively dispersing, and as a consequence, the speciation events for the host and the parasites are coincidentally the same age. But the host is not causing the parasite to speciate, the parasite is not causing the host to speciate. The host and the parasites are both fleeing from climate change, trying to survive. And as they disperse away and move around, they establish new associations. All of those associations are happening at the same time. Then you have isolation. The isolation events happen at the same time, so the hosts and parasites end up being the same age. So the story is actually far more interesting than a story of the host and parasite phylogenies mirror each other. It's much more interesting than that. And it's really interesting because the host and parasite phylogenies do not mirror each other. Because it's not co-speciation in the sense that I originally meant the term when I originated the term. It can still be co-speciation in the sense that you have everything running away together. And they're the same age because they were all running away from climate change together. And they established new associations. But this is not co-speciation in the sense that the parasites cause the host to speciate or the host cause the parasites to speciate. And it's not co-speciation in the sense of the host and parasite phylogenies being mirror images of each other, because they are not. They're not even close. And not even 50%. In all the classes I ever taught, if you got 40%, you failed the course. And so for this to show up in a textbook is either extraordinarily incompetent or unethical. Because Ridley was either too stupid to know that that's not a mirror image, or he didn't care. He had a story to tell, and he didn't care what the data actually said. And so I don't know whether he's stupid or dishonest. I don't care. But it's one of those. And the people who reviewed the book, and the people who reviewed the original manuscript for Nature, and the people who commented on it since then, are all culpable, because this is not co-speciation in the original sense. Nonetheless, it's an extraordinarily interesting finding. And all we have to do, and because they've got molecular clock estimates, because they were trying to make sure all the timing lined up, if those molecular clock estimates are robust, they have an estimate of when this all happened, which means that they can pinpoint these speciation events, speciation by colonization events, speciation by host switching events, to particular episodes of climate change in particular geographical areas. So if you do a biogeography study of this, you're not going to find bicarians. You're going to find a lot of dispersal. And this is the way a general phenomenon, this is a perfect example of how climate change can drive emerging disease. That's what this story really is. That's why it's extraordinarily cool. But it has been misrepresented and misinterpreted consistently, and now it's in undergraduate textbooks. And if you're an undergraduate and you're supposed to memorize this, and you're supposed to believe that those two diagrams are identical, that's tough. Because your eyes and your brain are saying, they're not the same. What am I missing? I have to study this harder because, this famous biologist who wrote this book says, these are the same. But my eyes say, they're not the same. What am I going to do? Oh, all right, I think I'll go to medical school. So, it's not co-speciation. Just like Don Rosen's classic case of vicariance isn't vicariance. It's cool, it's really interesting, but it's about sequential colonization. This is about sequential colonization. And it is a perfect example of how climate change can drive a whole diversification of emerging disease. That's what this is really interesting for. Okay, now, back to, so here's the new paradigm of the evolution of two different species. We can't even call it coevolution anymore if coevolution means mutual modification, or if it means mutually caused speciation, because none of that happens. And there's very little evidence of mutual modification except at very local levels. So, no, it's a new paradigm of something. Stuff living together. And the new paradigm is that there are macroevolutionary shifts from being specialists to generalists, and back to specialists and back to generalists, the oscillation hypothesis. And the way you get to be a generalist is, the first thing you do is expand your host range as a specialist through ecological fitting driven by climate change. Once you have a wide host range in a large geographic area, you then have localized adaptation, which means you go through a period of being a generalist, and then you have isolation and you become a bunch of descendant specialists. That's how that happens. You have ecological fitting, you have phylogenetic conservatism, not only in host resources, but also in parasite biology. You know, the most specific specialized aspects of parasite biology are also phylogenetically conserved. The part of the host you live in, the kind of life cycle you have, these are extremely specialized and extremely conservative in parasites. So the combination of host conservatism and parasite conservatism means that you have these extremely specialized associations with an enormous amount of flexibility if conditions change. That's ecological fitting. And this is driven, the diversification of these host parasite assemblages is driven by taxon pulses. So it's oscillation plus ecological fitting plus taxon pulses. And if you want to read a nice review article on all of this, there was a big review article published in Zoology in late 2010 on all of this stuff, with references by Augusta Janssen and some guy in Curso, some old guy that just put on the end. All right. Now, that means that if you're a parasitologist and you want to try to explain what you've got, you don't start with the host-parasite interactions. You don't start with parasites trying to kill their hosts, hosts trying to get rid of the parasites and all this crap. You start off with climate change. You talk about the interactions between hosts, because that's how you can share parasites. You talk about parasite evolution. You've got to know when in the phylogeny of your parasite group does something change that affects the way they get into different hosts. You might talk about how different parasites might interact with each other. One would expect in a situation like this that there would have been cases where more than one species of the parasite would end up in the same host, and yet today we find just a one-to-one relationship. What happened there? What kind of interaction was there between the parasites? And then you might worry about things like coevolutionary arms races or resource tracking or something like that. But this is part of what Thompson calls the coevolutionary mosaic. This is something that happens at very local levels that fine-tunes localized associations. But it's not something that gets amplified into large-scale patterns of diversification very often. It does happen, it just does not happen very often. And that's manifested in these very conservative phylogenetic patterns. Okay. Maximum co-speciation doesn't justify this in the view. The coevolutionary arms race doesn't justify that view. Okay. This new paradigm is what? It's just Darwinism. Ecological fitting is just a particularly interesting aspect of Darwin's necessary misfit. Or, as Deborah McClellan and I called it in the Nature of Diversity, Darwinism. See, we wanted to characterize Lamarckism and orthogenesis and Darwinism. So Lamarckism is every day and every way it's getting better and better. And orthogenesis is ye who enter here abandon all. And Darwinism was, hey, you can't always get what you want, but you can get one. And so, of course, one of the few reviews of the Nature of Diversity was by a Dutch philosopher who was extraordinarily upset with this. We were trivializing a major scientific theory. He was really, really pissed off about that. Now, if we had used some line from Shakespeare, he would have thought we were brilliant scholars. But because it was rock and roll, he was really upset. Which means our work was done. That's what we were trying to do. All right, so does any of this matter? Well, I might also say that another way of expressing this is what Harold Manter, a parasitologist, called parascript. So that parasites are simultaneously the product of a long history and of localized ecology. And in 1967, he published a paper saying, someday in the future, we may be able to figure out a way, a language, of allowing parasites to tell this story, this complicated story of history and ecology. And that story, the language will be called parascript. And so that's why Deborah McClellan and I call our book about historical ecology and parasites parascript. But it's just Darwinism. All right. If the host has the critical resource, the resource is a trait. If it's a trait, and if it's phylogenetically conservative, host switches are going to happen anytime there's a major climate change push. Anytime climate change makes macroevolutionary phenomena happen, you would expect a big outbreak of emerging disease. And all you need is climate change or species introductions or population explosion like humans. So here are some trivial conclusions. There's a lot of evidence for host switching. The theory of maximum co-speciation has been falsified in part by its own classic case. So we should get rid of it. Fortunately, we have a replacement already available, so you're not just abandoning ship and drifting on an ocean of uncertainty. And they know this too, because a lot of people who 5 years ago were saying it's all co-speciation are now publishing papers in which they say, we've just discovered a new phenomenon called host switching. No one's ever seen this before. All right, host specificity is an unreliable indicator of biocontainment. This is why every single attempt at biological control by human beings has failed. We have a perfect record with biological control. It's never worked. And the reason it's never worked is because nobody ever takes ecological fitting into account. Here's what they do. You have fruit trees in Hawaii. Suddenly you find that there's this insect that's gotten into Hawaii that's eating the fruit. And you find out that, oh, this is an insect that is a pest on these fruit trees in the country where those fruit trees are native. But they don't cause this much, they're causing much more destruction in Hawaii than in the native place. Why is that? Well, it turns out that in the native area, those insect populations are controlled by a parasitoid wasp. This is great. We will just introduce the parasitoid wasp in Hawaii, and it will control the pest insect. Have a celebration. We even have the governor of the state of Hawaii there to actually, open up the box and release the first set of hymenoptera and parasitoids. 2 years later, the obligate pollinators of those fruit trees are dying off because the parasitoids are killing them. Because the parasitoids actually like to lay their eggs in the pollinator insects more than in the pest insects. It's just that in the native range, the pollinator insects don't come into contact with the parasitoids. So what we do is we screw things up, and then we screw things up more, and then we screw things up more, because we have this overly simple view of the world. And we allow ourselves to believe that our theories are truth, and we never test to see. Nobody ever tests biological control agents in an evolutionary way. All they do is do a test to find out if this parasitoid in the lab will kill that pest. Yes, good, release. They never ask the questions, if we release that parasitoid in this new place, what else is it likely to attack? Well, nothing. It's highly host specific. It's co-evolved. It's not going to do anything. It's so specialized it will self-limit. That's what biological containment. That's what they mean by biocontainment. The parasitoid will self-limit. And they never do. Not only do they not self-limit, they go, banquet, look at all these new hosts. This is great. And so we always fail. We have always failed. We have also always failed with setting up conservation areas. The only thing we ever accomplish with conservation areas is that in some cases we actually accelerate local extinction rates. Good job. All right. What are the big questions in coevolution, or in the evolution of host and parasite association? The big question is something that's like a multivariate analysis of variance. Is how much of any set of associations has been mediated by biogeographic dynamics, that is, taxon pulses, climate change. How much has been due to host-host interactions, how much is due to host and parasite interactions, and how much is due to parasite-parasite interactions. We have to consider all of this stuff. Generally, all we do is count the number of hosts. And this is the real issue. If we're right, if the evidence that we have today that says all the host switches are episodic, then this is the paradigm case of how climate change can drive emerging diseases. A whole slew, an enormous diversity of emerging diseases. Which means that if climate change is going to intensify in the next 30 years, we have only begun to see the first and initial evidence of a global outbreak of emerging diseases in humans, in livestock, in crops, in sport fish, in food fish, wildlife. How many times do we see, is it once a week? We see some new fungus that's killing something, or some new something or other that's killing it. What if it's once a day? What if it's, we start listing the 5 new emerging diseases of the day? What happens? Well, it turns out that if we have enough of these emerging diseases, none of them have to be very deadly. But the cumulative effect of all of them is that it's going to collapse our economy. We'll talk some more about that tomorrow. I just want you to not be able to sleep tonight. I haven't been able to sleep for 10 years since I realized this. If I can't sleep, I don't want anybody else to be able to sleep. All right, so is this kind of a new beginning for studies of what we used to call coevolution? Okay, mutual descent, mutual association, not necessarily evidence of coevolution. Ecological fitting allows things to be really specialized, whether they co-speciate or not. Co-speciation may only mean mutual descent. It may only mean that they've been together in the same place at the same time, hanging out together. The old California model of coevolution. For real coevolution, we have to find evidence of mutual descent with mutual modification, and we have no cases of that. And maybe the reason we have no cases of it is because it doesn't happen. Under the proper conditions, host switching and other forms of ecological reorganization can occur rapidly and frequently. And they don't have to occur often to cause major effects in the biosphere. Now, we have evidence of this. I mean, this is just a very long, today is just a very long journey for me to get to the point with modern host-parasite associations that gives the same answer that the paleoecologists are giving us. And climate change perturbations can be very infrequent and yet can be the major driver of macroevolution on this entire planet. And one of the things we have to realize is that this whole emerging infectious disease business isn't new. It's not even new for humans. In fact, most of the cost of human civilization has been a sequence of emerging diseases. Every single parasitic disease that human beings have that does not have its sister species in a chimpanzee arose as an emerging disease as a result of something human beings did differently in their environments. That's why we have sheep parasites and cattle parasites, and pig parasites, and goat parasites, rat parasites. So this isn't actually anything new. And that's why the solution to the problem is not modern technology. It's not genomics and vaccines. How much money is being spent every year on developing a malaria vaccine? Okay. Why are we wasting money on a malaria vaccine? 75 years ago, there was malaria in Charleston, South Carolina, and it's gone. We got rid of it in the United States. We got rid of malaria without a vaccine. So we actually know how to get rid of malaria if we want to. So why are we spending billions of dollars on a malaria vaccine? Okay. Now, I'm very cynical, and I'm retired, so it doesn't matter. So my first cynical explanation is, it's because you can't get a Nobel Prize for ecology, but you can get a Nobel Prize for biotechnology. And the other cynical one is, we're not really trying to eradicate or even control malaria in places like sub-Saharan Africa because we care about the sick people in sub-Saharan Africa. We want a malaria vaccine in case we have to send 100,000 young Americans somewhere in sub-Saharan Africa tomorrow to kill people. And we want, it is the military-industrial complex that needs vaccines for these things because of massive species introductions. Every time, you know, I was teaching parasitology at the University of British Columbia in the early 1980s, my first faculty position. Students were doing term papers. This is in the early 1980s. So the students who are in the class weren't born until, you know, the mid-1960s. They weren't born until the mid-1960s. And this one student gives a term paper about, gives a presentation to class about the prevalence of malaria during the last 50 years. And she's going on and on. She says, now, you know, it looks like, you know, here we see in the 1950s, we see malaria really go down, but that was because of DDT. DDT is really bad, so we stopped using DDT. And we see the cases of malaria come right back up. But since then, they've been going down because we're using other measures. And it's a really nice paper. She's researched it really well. She said, now, the only exception to this is that here in the 1960s, in Southeast Asia, there's this increase in malaria, but the rest of it's just this nice. So she finished her talk, and I said, so what do you think accounts for that blip, that increase in the prevalence of malaria in Southeast Asia in the 1960s? And she goes, I don't know, tourism? And I said, have you ever heard of the Vietnam War? She goes, oh, is that when that was? So that's what's going on. The next time somebody sends a bunch of young kids into a non-native part of the world where they might acquire new pathogens through ecological fitting, somebody with a lot of money wants to have vaccines. Whereas, admittedly, that's a very cynical view. I'm claiming that every malaria researcher in the world really doesn't care about, you know, sick people with malaria, dying little 3-year-old kids with malaria, and I'm sure that's not true. I'm sure that at least some of them actually want to try to cure malaria. But the reality is we could eradicate malaria a lot cheaper. We took all the money that's going into biotechnology for malaria and we put it to work in basic public health measures. Now, how did we eradicate malaria in Charleston, South Carolina? Well, here's what we used to have. Here's the city, and here are the wetlands where all the mosquitoes are. Okay? And here's how the city gets water. Okay? How do you eradicate malaria? Use basic population dynamics. You cover these over so the mosquitoes can't breed in there. And then you drain this amount of the wetlands so the mosquitoes now, the distance between where the mosquitoes breed and where the humans are is greater than the cruising range of a mosquito. So the mosquitoes survive, the humans survive. But the only part of the biology that doesn't survive is the part that requires the transport. The malaria parasite. It just disappears as a demographic accident. It's ecology and evolution, it's not technology. All right. Tomorrow we have two topics. One is, I'm going to talk about a potential way to help integrate ecological and phylogenetic biogeography, and talk to you a little bit about how cool I think it is and why there's so much resistance to it. And then we'll come back and I'm going to hammer you some more about evolution and biodiversity and emergent diseases and things like that, because it's my last chance to pontificate in front of you. So we will see you tomorrow then. Thanks for your attention.